So even though I said I will combine two papers, I decided to separate them, even though they will be short podcasts. So this is another one that is a very nice study, but again, it depends on genetic analysis, and the details are kind of very technical. So I do want to include it here just to show how uh, evolution selection happens based on various things. We have focused on climate, but this is a good example of how a massive impact of a disease and the mortality due to that disease has impacted the evolution of immune genes in all of us living now. So let's just go through it briefly and try to understand what they did. Basically, they are looking at immune genes and other genes before the Black Death, during the Black Death, and after the Black Death, and look at their trajectories to see which of the trajectories changed, how the genetic frequency of certain genes, immune genes, got selected because of the Black Death based on this technique. Okay, as simple as that sounds, the techniques involved are very detailed. Infectious diseases are among the strongest selective pressures driving human evolution. This includes the single greatest mortality event in the recorded history, the first outbreak of the second pandemic of plague, commonly called Black Death, which was caused by the bacterium Yersinia pestis. This pandemic devastated Afro-Eurasia, killing up to 30 to 50% of the population. 30 to 50% of the population. So those who survived carry the impact of the disease, and that's what leads to the long-term impact on genetic selection. To identify loci that may have been under selection during the Black Death, we characterized genetic variation around immune-related genes from 2006 ancient DNA extracts stemming from two different European populations before, during, and after the Black Death. This is very critical. To understand the impact of Black Death, you need to know the loci of these genes before the Black Death, during the Black Death, and after the Black Death. Immune loci are strongly enriched for highly differentiated sites relative to a set of non-immune loci, suggesting positive selection. We identify 245 variants that are highly differentiated within the London dataset, 4 of which were replicated in an independent cohort from Denmark and represent the strongest candidates for positive selection. The selected allele for one of these variants, RS2549794, is associated with the production of a full-length versus a truncated ERAP2 transcript, variation in cytokine response to Y. pestis, and increased ability to control intracellular Y. pestis in macrophages. So a lot of technical terms here. Allele, you know what that is. Basically, gene is what gets expressed. So from the parent, you get two genes, let's say for hair color, and one of them gets selected, but the two, when you inherit them, are called alleles. Okay, so one of them gets selected, obviously, and ERAP2 transcript is basically a response in T cells and the immune response in the body in responding to perturbations or attacks from viruses like Y. pestis. Okay, and macrophage is a cellular level function that identifies the invader and tries to fight them. Okay, so that's the simplest explanation I can give. Finally, we show that the protective variants overlap with the alleles that are today associated with increased susceptibility to autoimmune diseases, providing empirical evidence for the role played by past pandemics in shaping present-day susceptibility to disease. So, to fight the Black Death, the human body did something and all those who survived because of this selected immune response, ERAP2 transcript and so on. And that then came with additional impact, and that is the susceptibility to autoimmune diseases. This is very relevant because we are sitting here as I'm recording this in October 2022 with a few million deaths from COVID-19 and lots of impacts still going on in terms of the long COVID, and a new variant has emerged, and there are lots of warnings from the World Health Organization and countries like US saying this winter could be another outbreak of COVID-19, and China is still putting a lot of people under lockdown with their zero COVID policy. All this mess and the way the disease went around the world, did it do anything to the immune gene or its locus going into the future, and what will be the side effects? So there are already many research or data based ideas that say that COVID has had an impact on various things, including heart and brain and so on and so forth. So this disease pressure on diseases as selection pressures are critical. So just to conclude, look at the population in thousands before, so start at 1000 AD, Anno Domini, or you can call it Common Era, and then there is a population boom before the pandemic, the Black Death, second of them. The first one doesn't show up here. This is a very schematic thing anyways, but we came out of the medieval warming and got into the Little Ice Age starting about here, but we will not discuss what may have caused this. And then the Black Death hit around 1350 or so and lasted for decades, and it crashed the population by 30 to 50% in the Afro-European regions, and then the population obviously recovered. But this massive impact on the population numbers and distributions, and those that survived through the Black Death, carried the impact in terms of the immune genetic selection. Okay, it's very directly related to how we have evolved. I have one podcast on how past diseases have had an impact on our evolution. This is a clear example of how seriously it has left the impact on immune responses, the genes for immunity. We have also talked about Neanderthals having left pieces of their DNA in all the modern humans. So all these things are kind of interesting but not necessary to the main story of climate and evolution, but I'm going to include them here as new findings just because they are interesting and related. Okay.